Good morning, everyone. I'm here today to tell you about the hybrid pondweed that is in Otsego Lake. This plant appears to be a hybrid of a worldwide native plant and a North American exotic plant. We believe that the parent plants are the native clasping-leafed pondweed and the invasive curly-leafed pondweed. To your right here, you can see a picture of the fact sheet of this plant, and this is available on the OLA website for you to peruse at your own leisure. Above it are some photos from my time in the lab. Of particular interest are the two in the middle, and this is the same individual, but I put the picture in half to try and show you guys the length of it, because this is one of the longest individuals I have found this summer. And the length of this individual leads me into my next slide. So when it's just one or two of those long strands, it's not so much of an issue, but when they form dense beds, as you see in this picture below, that's when it becomes a problem, because these dense beds inhibit recreation and have a potential to impact property values in the long run. Additionally, this is the fourth year since the plant was first noticed in Otsego Lake and it's been spreading. I've seen dense beds down by the lakefront boat launch, as well as by the river outlet and the golf course. I've seen clumps or patches going up both sides of Otsego Lake, even past the BFS main lab and boathouse, up to Hyde Bay and residential houses. I've even seen it as far north as Springfield Landing. So this plant is definitely well established in the lake. So my study has been focusing on sampling for insects that could be eating the plant, in addition to documenting plant density and heights. I have also been mapping waypoints of plant beds or smaller patches. So up here is a picture from one of my days in the lab, and you can see numerous types of caterpillars that I found on one of the plants. And then over here is an example of the top of the plant that's in very good condition up here, but as you work your way down, you can see it in various stages of decomposition. And I included this because a lot of people have been seeing the plant, and originally it was in good condition, but as time has been going on, some plant beds are not in good condition. So I just wanted to include this as an example. In terms of the herbivore results, we collected samples from the 30th of June and from the 7th, 14th, and 21st of July. A variety of herbivores were found on the leaves of the hybrid pondweed, including midges, which you can see one pictured here to your left. I also found caddisfly larvae and damselfly larvae, and sometimes cases holding them, or different cases such as egg sacs. I found a number of caterpillars and snails, as well as a few leeches during my time analyzing those results. Here is a picture of some bits of mud, and it doesn't look like much, but sometimes the midges would hide in these little houses over here. And at one point I found about 8 to 12 in all these little mud clumps. So I thought I'd include that as a picture of what I found. And then here, this is a mayfly, one of the organisms I found. This one is a whirligig beetle. This third specimen over here is a midge. If I remember correctly, this organism up in the top left is a caddisfly. And then over here in the middle we have a variety of snail shells and a scud. This is a worm of some kind, and then this is one of the few leeches that I found. So as you all can see, there were a variety of organisms that were found, but overall the most common herbivore that I encountered was the midge, followed by the spired and flat snail shells, and then the caterpillars and scuds, and then the few caddisflies and damselflies at the end. Over to your left, you'll see a picture from one of my Excel sheets, and this is how I keep track of my data. I would enter the coordinates and the waypoint name, and then I would take the length of the plant in centimeters and count the number of leaves and stems that it had, and look for any signs of herbivory. If I had any signs of herbivory, I would place the stem in a bin of water and shake it around to knock loose any mud or clumps of dirt like you saw on the other slide, and then I'd look for different herbivores. And here, as you can see, I found a seed of some plants, some egg sacs, several midges, a scud, and so on. For mapping waypoints, I've been using Garmin Basecamp and QGIS to document the location of plant beds or smaller patches. And then I put my waypoints on a contour map of the Otsego Lake, and this is to try and determine the depths as to which plants are growing. And a map such as this one to your right here will allow students in the future to return to sampling sites. Additionally, this allows me to keep my data organized and allows me to add more sites later in the summer if I so wish. For ongoing studies, as of now, I'm starting to use PERTRAM, which is Point Intercept Rake Toss Relative Abundance Method. This allows us to determine the percentages of plant species and the size of them at different areas. So up here at this picture, I used Microsoft Paint. And what I did with one of my original waypoints was I zoomed into it and then formed a grid by numbering all these different boxes. And at each interval, you take 3 rake tosses. In the upper left here, this is a sample from one of my rake tosses, and then down here is the second one. And this is a smaller one, but I was still able to separate the hybrid pondweed from what I believe appears to be stonewort there. And I just write down the different percentages of the plants that make it up that sample, and then I'll be inputting that data to Microsoft Access to analyze it soon. In terms of a solution, I'm honestly not sure as to what the best route is as of now. As I've been sampling the past few weeks, I realized that a few of the plant beds are not in great condition, and we are trying to determine why. We were wondering if someone was potentially using herbicide illegally, or if it was due to high insect activity, or if it was something with the water temperature. When I was sampling yesterday, in one of my rows on one of my grids, I saw that the plants near the shore were not in good condition, but those that were in deeper waters were in much better condition and appeared to be healthier. So I'm going to continue to monitor the changes to the plant beds this summer, and I'm going to see if that trend continues for the rest of that grid that it was in and for any other grid. Additionally, I believe that monitoring should continue for next summer, especially by lakefront near the golf course and the outlet, because these are the newer beds and they're very dense. As I'm drawing to a close here, I wanted to say a big thank you to the Biological Field Station and the Otsego Lake Association. Without both of you, this project would not be possible, so thank you for helping to organize it and for funding it. Truly, thank you so much. Also, a big thank you to Paul Lord for being my mentor and teaching me how to drive a boat and not crash it into anything. We're getting there. And also thank you to Lord and Sarah Coney for helping me to identify the herbivores and just helping me to learn more about the lake. Additionally, I wanted to give a shout out to Holly Waterfield. That contour map you made of Otsego Lake was awesome and it helped us to stay on schedule, so thank you so much for putting in your time to give that to us. If anyone has any questions or concerns, please feel free to contact me at my Gmail or at my LinkedIn, and thank you guys so much for your time. Have a good day.